Instagram goal.
It's, yeah, I know, it's,
Okay, you're good.
you good?
Yeah.
Okay, take two. Good morning, everyone, and Hannah and Maddie, and Ailey. We're gonna be talking about olive oil today, and I'm not gonna rehash any of the jokes I just said because that would be weird for everyone here. But to start off, I have a video for you all. Next best thing.
Dentro terra ci devi stare per capire quello che è, se no non lo capirai mai più mai, perché c'è una nozione, se no non ci sta niente da fare, ma chi non l'ha passata non l'ha capito. E a me piaceva, e mi piace ancora. Allora la mattina ti alzavi e andavi tutto in campagna, e dopo si mettevano i teli e si mettevano le scaglie di legno. Le scaglie di legno, ognuno aveva quella sua. Era la soddisfazione mia a tirare giù l'oliva. La sentivo proprio sopra i teli, tutto quello che scendeva l'oliva. Era bello, guarda, qualche cosa di bello. Ogni quintale d'oliva raccolta ci fai 20 litri d'olio, che hanno un quintale c'è una giornata, specialmente quando ci stanno le gelate, la mattina, la notte gela, quando tu vai a prendere l'oliva, se si spaccavano le mani, e la campagna è fatta così. E vedi quando che esce l'olio, esce verde scuro, e senti un profumo che è qualche cosa di buono. Ma dopo te lo mangi pure. E quando te lo mangi, adesso ti do un segreto, quando ti vuoi sentire l'olio buono, ti fai una fetta di pane, prima metti il sale, poi metti l'olio. Ora il palato sente subito il sapore dell'olio. Uno raccomanda ai giovani di mantenere questa cosa dell'olio, perché è un toccasana e fa bene. L'olio mio è come esce dall'oliva, non sprecatelo, però usatelo per la necessità, perché l'olio è buono, è l'oro, è oro proprio. È un regalo che gli fa nonna proprio, è un regalo che gli fa nonna Nerina.
I know.
Cheering up.
So, Nonna Nerina called olive oil a cure-all. I don't exactly know if it's a cure-all, but it has some really cool properties. The focus of today's presentation is going to be on how olive oil can regulate intestinal inflammation. And so we're going to be talking a little bit about the pathophysiology of inflammatory bowel disease, because not a lot of people care about intestinal inflammation until it actually presents itself as IBD, right? So it can be lurking in a subclinical state, increased intestinal inflammation, all of a sudden a combination of things occur and you have disease expression in either ulcerative colitis or Crohn's disease. And so we're going to talk about how some compounds found in olive oil can help modulate these inflammatory responses and potentially restore proper function to the body. But first, we need to understand a little bit of what's going on in IBD. What's the pathophysiology? How does it work? So, what I need you guys to know, it's a combination of things. It's not one thing that causes IBD. I can't just poke you and boom, you have IBD. It's a combination of your genetics, your gut environment, and your gut microbiota. And these all playing together. Now this picture over here gives kind of a good idea what's going on. over here is kind of like a healthy gut. Over here is your diseased gut. And there are two big things that happen. The first thing is you have a loss of epithelial barrier integrity. So you have these tight junctions here. Tight junctions hold your cells in your gut like this. So nothing can go through between them. If someone wants to get from the intestinal side of the gut into the blood, they have to go through the cell. In IBD, those tight junctions get loosened. Things can slip through, you generate an immune response. You also have tissue destructions. You'll see in some histological pictures I have later, these cells get all, like, they lose their shape, they lose their structure, and thus their function. Lose your structure, lose your function. And lastly, there's a ton of inflammation, because when you start destroying things, the body comes in and goes, hey, we need to fix this. And so there's an inflammatory process. Now, the inflammatory process in IBD is a misapplied inflammatory process. It shouldn't be there. It's there for a reason. It's hurting the body. And so we see upregulation of a whole bunch of inflammatory cytokines. We'll talk about some of those later, as well as a further immune response that ultimately leads into IBD. So I need to, that's actually the next slide. Olive oil polyphenols are the compounds of interest today. You find them in pretty much all parts of the olive plant. But we're going to be talking about specifically the olive leaves and the olive oil. That's where they're found in the highest concentrations. Extra virgin olive oil is special, okay? Virgin, the word virgin means unmixed. So extra virgin is oil that is extra unmixed, okay? So when you first press oil in, you take your 2 stone slabs, you put the olives in it, that first oil that comes out, there's been no heat applied and no other chemicals applied to extract the oil. You keep your phenolic compounds in that press. The moment you add heat, or the moment you try to add a chemical to pull oil out, the phenols are really, really just delicate, so they get destroyed. Okay, so you're only gonna find the polyphenols in extra virgin olive oil, or if you've done another method of extraction to pull those polyphenols out and preserve their integrity. Once they go into your body, they go into your gut, they come down into your stomach, pop out through the duodenum, and then what happens? They go right through the hepatic portal system. As our favorite function of the liver that we all know and love is first pass processing. So the compound goes into the liver, it gets changed, and then gets spit out the other end. Unfortunately for us, once those compounds are processed, they again lose their function in the gut. So we can't use them. But as in all biological systems, there's inefficiency, and that inefficiency actually works to our favor here. If those compounds aren't taken up for first pass processing, they go right down to the small intestine where they end up in the colon. Once they get in the colon, that's where the magic happens. So if you take enough polyphenols and you can skirt them past first pass processing, as they get excreted, then they begin to act in the colon. And once you get into the colon, you can have some of the molecules like allerapin, they get modified by the gut microbiota to form things like hydroxytyrosol, which is also a polyphenol. I'm going to introduce that to you in a second. And that does a whole bunch of really cool stuff in the gut. So you just want to be taking higher concentrations of polyphenol. The theory anyway, and we'll show how that might be the case, is that if you take enough of these polyphenols, you get them into the gut in a high enough concentration to get them to do the things that you want. So I'd like to introduce you to a couple of my good friends, the olive oil polyphenols. First is oleocanthal, and this is what he looks like. Another one is hydroxytyrosol, and that's what he looks like. And lastly is allerapin, which is made up of hydroxytyrosol and a couple other things. These are the three that we're going to be focusing on today, so just kind of remember their names. You don't need to remember their structure, but they have a phenol ring, which is why they're called their phenols or polyphenols in the case of allerapin and some of allerapin's buddies that are beyond the scope of this presentation. You find them in the fruit and the leaves. It's an important point I'm trying to make. Okay, so what happens in that IBD pathophysiology that we talked about earlier once you add in olive oil? So this here is our overview slide. I'm going to tell you all about the stuff here, and then I'm going to come back in a little bit more detail and show you some of the research behind that. So you have your IBD, intestinal epithelia. What happens? You intake your olive oil that has olive oil polyphenols. What's one of the first things that it does? Well, it increases gut microbiota diversity. How does that happen? Polyphenols are really interesting and difficult compounds to digest, and so only certain microbes are able to do it. You feed the certain microbes, these polyphenols, they grow in number. Turns out, if you're a specialized microbe, very generally in the gut, and you're only able to digest certain complex molecules, you actually, on a broad speaking, have a more beneficial impact on the host than if you can just digest everything. Those are the guys that cause all of your infections, E. coli, salmonella, so on and so forth, generally speaking. They also have some immune action on the lymphocytes. They'll inhibit that, so they'll decrease lymphocytic activity, which is great if you have an uncontrolled immune response. And important for us in that tight junction physiology, remember we lost that tight junction, right, capability, so we had the leaky gut. The polyphenols also act on the ZO1 protein, which is attached to globulin, and that helps those tight junctions stick together. Okay, so we've been able to stick our gut together and begin to repair it, so now things can't pass through, they have to go through the cells. But the cells are all inflamed, so what are we gonna do in there? There's a whole bunch of metabolic action that the polyphenols have. They inhibit iNOS, they inhibit COX-2, they decrease reactive oxygen species, which downregulates the P38 MAPK pathway, which has a whole bunch of really cool stuff here in the nucleus, ultimately downregulating pro-inflammatory cytokines. Like, there's a lot going on in here. Don't worry, I'm not going to make you memorize it all. We're going to talk about a couple of these pathways in a little bit of detail. So, let's look at this from a cell culture perspective. Let's look at this and take, okay, we're going to take maybe the gut of somebody who has like IBD. We're going to take it out of their body. We're going like with a biopsy. We'll culture those cells, and then we'll do experiments on them. Right? So this is called an ex vivo, out of life, right? Out of the body. You take it out, and you do these experiments in cell culture on a dish. So we're going to move down to animal and human models later. So, in cell culture, what we're going to try and show is that olive oil polyphenols have an effect on inflammatory mediators that are present at a cellular level. And so there's pro-inflammatory molecules, I split them up into two groups. Pro-inflammatory cytokines, TNF-alpha and IL-17 are the two we'll be talking about, but there are others, like as IL-6, IL-1 beta, and pro-inflammatory enzymes, COX-2 and iNOS. So COX-2 is cytoxygenase, we all know and love this one, and iNOS is the inducible nitric oxide synthase enzyme. And so these are all involved in creating an inflammatory response in the body. So I'm sure you guys know some very common COX-2 inhibitors. Does anyone happen to know off the top of their head? Go on, say something.
No.
No? Okay. Advil. Ibuprofen is a COX-2 inhibitor, so it will actually directly act at location to decrease inflammation. That's why it's an anti-inflammatory. Acetaminophen's not, it acts in the thalamus. So you pop your Tylenol all day, it's not going to decrease the swelling in your foot, but if you take your Advil, it's actually going to lower inflammation locally wherever you might have that issue. That's also why it works for menstrual cramps. Ginger is another COX-2 inhibitor. It has a molecule that acts exactly the same as ibuprofen. So if you've ever been sick and you've taken ginger and wow, I feel way better, one of the main mechanisms of action is by inhibiting COX-2. So, in this diagram here, I'd like to show you that allerapin also is a COX-2, maybe not inhibitor, but reducer. It has an effect on the activity of the enzyme. But in order to do that, we need to remember some of our friends, the gram-negative bacteria. You remember inside gram-negative bacteria, you have an outer membrane, which is composed of lipids, a little bit of protein, and some oligosaccharides. We call that the lipopolysaccharide, the LPS. that is incredibly, incredibly immunoreactive. You put a little bit of that in anywhere with an immune system, and like the sirens go off, the lights go off, massive, massive, massive immune response. So let's take some of that and shove it into our ulcerative colitis patients and see what happens. So here's our control. Beta-actin is also a control. And so what we did is we grabbed some gut samples from ulcerative colitis patients, and we put them into a dish, and we challenged them with lipopolysaccharide. Okay? And so this is what we did here. We see from, if you didn't put anything in, you put your lipopolysaccharide in, you have an increased amount, or I guess response of amount, or immune response, of COX-2 expression. And you can see this in the diagram here. The line is darker over here. Okay? But take a look next door. If you add Alerapin, you actually reduce that response. So this is definitive evidence here that there is some response, that Alerapin does actually reduce COX-2 expression in the presence of an inflammatory agent, so it acts as an anti-inflammatory. One more, same thing happens here. IL-17 is also reduced by Alerapin. Again, have a look and compare our Alerapin versus our control. But you're probably asking, Matt, do we actually have any numbers to this? Because you're presenting data that are in arbitrary units. That's what AU stands for. And yeah, I understand that, right? Whenever you do Western blotting, it's always in arbitrary units, always. Because you can't exactly quantify. The only way you can quantify this is by shining a light and going, how much of the light was absorbed, how much was not absorbed. There's no numbers to it. Luckily, if we head over to the lab next door, we can throw an ELISA together and we can actually see, so an ELISA is a way you can actually measure specific protein concentrations, and so that's what they did. You can actually get a number on this, and you can show that Alerapin actually can reduce by about 20 picograms per milliliter the expression of IL-17 in an ulcerative colitis patient who has been challenged. So this is your standard ulcerative colitis patient, right? Actually, sorry. Yes, it is. This is the standard ulcerative colitis patient. This is when you challenge it with increased inflammation, and you're actually able to bring that UC patient back. Now, these two, they didn't run any stats, so you can't say if it's significant or not, but you're actually able to bring them back to about the same level as you were before you introduced that inflammatory kick. And so, same people, same patients, like this is the micrographs of the cell culture. And so you're actually able to see increased organization of the tissue when you've challenged it when you've protected it with Alerapin versus when you've just challenged it with LPS, and decreased T lymphocyte infiltration in that tissue when you've protected it with Alerapin, which again further goes to show that Alerapin does show a protective effect against inflammation, so it's an anti-inflammatory effect. Here's some further evidence from cell culture. You see in a peritoneal mouse macrophage model, again when they challenge it with lipopolysaccharide and then protect it with extra virgin olive oil extract, you see an inhibition of intercellular reactive oxygen species, nitrate production, again, decreased COX-2, as we mentioned before, and downregulation of the iNOS protein. And then they further they further went into that and said, how did this happen? Well, it was because we downregulated this other pathway called the NF-kappa beta and the MAPK pathway. And so those are just pathways that induce COX-2 and iNOS. In another cell culture model, they looked at intestinal T cells, and they showed that extra virgin olive oil extract promoted apoptosis and reduced T cell activation. Remember that reducing apoptosis thing. We're going to come back to that in a little bit. In another mouse macrophage model, they did a very similar thing. They exposed it to LPS, but instead of giving Alerapin, they gave hydroxytyrosine, and we also saw the exact same inhibition of the NF-kappa beta pathway and reduced inflammatory cytokine expression. Lastly, yes, lastly from the cell culture, we saw, so remember how over here we said we had this apoptosis thing, the promotion of apoptosis? Well, they also found that if you took these phenols and you put them into a cancer cell model, right, and this HT29 is an immortalized cancer cell model, and then you treat it with hydrogen peroxide. So hydrogen peroxide is like everybody's favorite, we want to give you cancer thing, because it's a free radical and it will just go, that's why it's so good at wounds, right? It will go in and it will just tear things apart. Tear DNA apart, tear your whole cells apart, tear your lipos, like everything goes crazy with hydrogen peroxide. Whole bunch of free radicals. So you can use it to induce cancer, like a carcinogenic effect in a tissue culture model. But if you protected it with the olive oil phenols, you saw anti-genotoxic effects. You had to pre-treat them, you protected them, but you saw increased barrier function of the calco2 cell line, so the cells held together better, which is what they're supposed to do because they're intestinal cells. Normally, if you treat them with hydrogen peroxide, they all break apart, but they were held together if you had the polyphenols in there. And they also saw decreased cell invasion and attachment, which is another feature of cancer cells, as well as the metabolites from hydroxytyrosol, like so the colonic metabolites are the metabolites from the polyphenols. They actually can initiate cell cycle arrest and apoptosis. And that's one of the big issues in cancer cells. The cells don't stop dividing. But if you can initiate cell cycle arrest, if you can tell the cell, hey, you're damaged, die, you're able to stop cancer before it starts going crazy. So, additional evidence from cell culture that olive oil polyphenols have anti-cancer properties. Summing this up, wrapping this up, it's clear that olive oil polyphenols are able to affect major inflammatory pathways. These aren't minor ones, these are major ones. These are the big ones that get lots of airtime in the human body and animal physiology as well. P38, MAPK, NF-kappa beta, and INOS. We also saw anti-cancer effects and antioxidant effects. So, olive oil and its polyphenols in vitro, and ex vivo, in a cell culture model, can modulate several key points in the IBD pathophysiology. Now, let's move on to animals. So in animals, a couple things this is like a big kind of, again, like same thing, we had the overview before, now we're gonna have a big overview again. In animals, here's what we found. We found that, and I wanna look at this stuff down here, right? If you treat with olive leaf extract, you see a decrease in pro-inflammatory cytokines, these as well, pro-inflammatory inducible enzymes, adhesion molecules and chemokines, so we're not gonna get too much into these, but these two guys affect the ability of the cell to migrate and move somewhere else, so these are your cancer properties, and these affect your epithelial barriers. So we're not gonna be talking about these, we're gonna be mostly discussing the anti-inflammatory effects, but it's important to know that there are anti-carcinogenic effects that we haven't fully done a deep dive in here. Yes, so INOS and COX-2, keep those in your head, we're gonna be talking about those. Because in rats that have induced colitis with DSS, DSS is a chemical that we use to induce an inflammatory response in a chemical, or in a gut. It's like the gold standard for inducing an inflammatory response to mimic IBD. We see that extra virgin olive oil actually attenuates that response. So have a look over here. You'll see in the COX-2, when we protect with extra virgin olive oil, we have a significant decrease in the COX-2. So this is a COX-2 to beta-actin ratio. So this is saying, okay, here's your control. This is what happens if you don't do anything, right? And this is what happens if we are looking for COX-2. And now we're gonna standardize these to each other and then take a ratio and say, how much did this or this change from this? How much did this change from this? How much did this change from this? And so, yeah, you're saying, well, what's going on here? You know, this looks different from this looks different from this. Well, don't worry, because they're all standardized to each other. So they were all run in different wells, perhaps at different times. And so if one run produced a stronger signal, that should be amplified in the entire gel. right? So you're not too concerned about these being different from each other, but you're really looking at the comparators between the COX-2 and the beta-actin because they were run at the same time. And you see the same thing with iNOS. There is a protective effect when you induce colitis and when you protect with extra virgin olive oil. And we see that again in our tissue samples. Look at the control tissue samples. They're very beautiful, very pretty. There's lots of order. You're very able to clearly see your intestinal epithelial cells. And then we just go and ruin it all when we induce an inflammatory state. Like, they're all gone. There's very little order here and none over here. But we can actually recover a little bit of that. So this is the proximal, middle, and distal colon, right? So different segments. We can recover a little bit of that. Not all the way back to, like, perfect, because we saw in that last paragraph that last bar didn't go all the way down. But we actually can recover. We can protect from damage, which is really, really neat. This isn't an animal model, this is in rats. There are some further studies that were done in animal models. We showed, there was a study that combined an in vitro and an animal model, and what they did, they were looking at medium chain triglycerides, oleic acid and hydroxytyrosol, as well as fish oils. And so medium chain triglycerides, they got a lot of airtime with coconut oil a couple years ago. And they said, okay, let's just throw all these oils at our animals and at a cell culture model and see what happens, right? And so that's what they did. They took their little dishes of their in vitro models, and they said, okay, we're gonna put all these oils on and see what's changing. So when they put the olive oil on, so the oleic acid and the hydroxytyrosine, that actually reduced, and so T-butyl hydroperoxide is another damaging molecule. That reduced damage on the cells, whereas if you put medium chain triglycerides, you actually got more damage. And then, if you put fish oil and olive oil together, there was a protective effect, synergistic. So they worked better together than each of them did apart. And you also had decreased inflammatory, like IL-8, so inflammatory molecule production, cytokine, inflammatory cytokine. And so then they confirmed this. This was in cell culture. They confirmed this on a rat. And they found the exact same thing. The fish oil and the olive oil combination in an animal had the exact same synergistic effect that it did in the cell culture. This actually proves it 1-to-1. Hey, you can take a cell culture result and draw a direct line to what happens in the animal. Cool, eh? And MCT oil had the exact same response. It increased inflammation. It exaggerated the inflammation. So this is a pretty powerful study to show that the results you make in cell culture, you can actually bring them to the living model, and the premise holds. They did another study where they just gave rats extra virgin olive oil. They had a control rat, they had a standard, rat chow, extra virgin olive oil diet, and they saw that the olive oil diet attenuated a bunch of these inflammatory factors and decreased cell proliferation and increased cleaved capsase 3 levels, again, anti-cancer, anti-inflammatory properties. So what can we learn from those animals? Well, we can learn that the animal studies pretty much confirm the evidence that we found from the in vitro studies, and that they act in the same manner. And we see some common effects, such as suppression of inflammatory pathways, improving the histopathological signs. The disease activity index, I didn't touch on in this presentation. It's in the paper that I based this presentation off of, and the anti-cancer effects. Okay, so now we're going to look at humans. In humans are kind of tricky to do studies like this on, because you need to get biopsies, and people don't like giving biopsies. So you're not able to get a ton of statistical power. And you can't take people and just do what you would do to, rats on them, right? That's not ethical. So we have to find different ways around that, right? And different ways that we can structure our studies to still get the same information. So the first big study here was a randomized crossover clinical trial. Crossover trials are the absolute, in my opinion, the best study you can use on humans. Why? because randomized controlled trials just do not work very well when you're trying to make sure that you control for individual differences. We're in an era of personalized medicine, and in an era of personalized medicine, we need to be able to tailor an intervention to you, to you, to you. What Derek gets is not going to be what Diane gets. It's going to be tailored to Diane's unique physiology, what's going on in her body at this time, right? And so in a crossover trial, you are your own control. So you do the one study, you have a washout period, and then you get the other intervention, and so you can compare your results to your previous results. And so that's exactly what they did. They gave extra virgin olive oil, they gave canola oil. And they found that, after this washout period and the whole study was completed, that when you were taking olive oil, you had a decrease in ESR, erythrocyte sedimentation rate. And you also had a decrease in C-reactive protein compared to canola oil. And then you had a whole bunch of symptoms that were reduced, such as bloating, constipation, fecal urgency. Right? Remember, these are ulcerative colitis patients, so they have all these symptoms, and olive oil attenuated those symptoms. It actually helped them, practically, as a person. Good morning, Alonzo.
1 minute.
All right, we'll finish this up. So, here is some of the data from their studies, and don't worry about the big table, and you just need you to look at those two numbers there. So you'll see erythrocyte sedimentation rate and C-reactive protein, both were statistically significant. They had a decrease. So this was their change here. They had like minus 0.18, right? Compared to their canola oil buddies over there. Now, if you're paying attention, the astute viewer might wonder and say, hey, Matt, didn't we say that TNF-alpha had a statistical significance, like a decrease in animals? But you just showed me over here that in this study they didn't find it. What's going on? Where's all your, the whole, big olive oil, this is done, right? We've just blown a hole in your whole presentation. Well, remember what our animal intervention was, right? Oops. Our intervention was olive leaf extract in animals, in this big study here. In the human study, they gave them extra virgin olive oil. Olive leaf extract has a significantly higher concentration of polyphenols than olive oil does. Way higher, like loads, bounds higher. So if you're taking a lot more of those, you're gonna have a higher concentration that end up in the gut. If you have a higher concentration that end up in the gut, you're gonna have a higher effect. You have a stronger effect in the gut. So you can't blow off the whole thing just because the human study gave all extra virgin olive oil and didn't see the results. But that is definitely something noted that I noted actually, and it wasn't talked about too much in the paper, was that there wasn't a lot of homogeneity within the studies. Some were giving extra virgin olive oil. Actually, we're going to talk about this in the discussion section, but some were giving extra virgin olive oil, some were giving olive leaf extract, some were giving polyphenol enhanced olive oil. And it's like, okay guys, we need some standardization here. But it's nice to see. Here's a big study. This was a cohort study. You want your numbers, here they are. 25,000 people, an EPIC study, and oleic acid showed to be a protective factor for ulcerative colitis incidence. So if you had a lot of oleic acid, if you were eating a lot of if you were eating a lot of oleic acid somewhere, you had a much lower risk of developing ulcerative colitis. It protected you. So, in discussing this, what did we learn from the human studies? Well, the human studies, we see the same effects that we saw in the animal studies, some of them, and the in vitro studies. We see anti-inflammatory effects. ESR and CRP, those are global inflammation standards, because you can't look at TNF-alpha on a human, because you'd have to go in and take a biopsy. People don't like that very much. But you can take a blood draw and see ESR and CRP. So you can make that correlation that there is an anti-inflammatory effect on the whole body just by having olive oil. They also saw symptom reduction, which is some of the things that they also saw in the animal studies. We saw that kind of in the histopathological side, as well as in the studies they talked about how the animals would just get diarrhea when you inflame their colon, and how it would be reduced when you give them the polyphenols. And they also saw microbiota changes, and these are some other studies that I didn't present due to lack of time. We need to be careful, and so this is what I was mentioning earlier, we need to be careful in making a one-to-one correlation that everything that happened in the cell culture model can happen in the animal and the human model, but we have pretty good evidence that it at least goes from the cell culture to the animal model. And if you get it to the animal model, you can show that in an animal that this stuff happens just as it did in the cell culture. It's much less of a leap to say that it happens in humans as well. Because animals model humans way better, like it models a living system way better than just an ex vivo cell culture model. So in conclusion, yes, olive oil polyphenols can modulate inflammatory state in the intestinal lumen. Should you be drinking quarts and quarts and quarts of olive oil? Well, I'd recommend just going for olive leaf extract, right? Because you're still getting that content of Alerapin. And here's just some commercially available brands that provide Alerapin. Now, I found it interesting to note that like the Now brand, which is kind of like your cheaper brand, 6% Alerapin, but if you get into, you know, the True Hope brand or this Health's Harmony brand, right, they got 20% Alerapin. So you're getting way more bang for your buck. You're getting a lot higher concentration of Alerapin, which, as we learned, you need that high concentration to get through the first pass processing so you have enough in your gut to do what you want it to do, because the response is dose dependent. So I hope that you learned something about olive oil and the polyphenols, and maybe a little bit about how multi-level research is conducted through cells and cell cultures in animals and then into humans, and can apply that in your practice one day. Be it olive oil, or be it you're just trying to review literature and you're like, man, how do I take what I learned in the cell culture model and apply it to the animals and the humans? And thank you very much for your time. There are a lot of papers at my desk.